This is Lecture 34 on the development of the brain. During week 4, while the embryo is folding, this causes the brain to bend ventrally together with the head fold and ends up producing these brain flexures. And there are 3 main brain flexures: the midbrain, the cervical, and the pontine flexures. The cervical flexure forms the delineation between the hindbrain, the most caudal part of the brain, and the spinal cord. And the pontine flexure forms a separation between the 2 different parts of the hindbrain, which are the metaencephalon and the myelencephalon, which we'll get to in a minute. And here we see a figure of the brain at an early embryological stage, and here we see the midbrain flexure, this bend right here. We see the pontine flexure as well, and the cervical flexure. And as I said, here's the level of the cervical flexure. So above this is the brain, and below this level is the spinal cord. And this is a really good figure of the development of the brain vesicles. So early in development, we have a neural tube which differentiates into the 3 primary brain vesicles: the prosencephalon, which is the forebrain, the mesencephalon, which is the midbrain, and the rhombencephalon, which is the hindbrain. As you progress further through the development, 2 of the primary brain vesicles further differentiate, so you have a total of 5 secondary brain vesicles. These are the teleencephalon, which is the most rostral part of the forebrain, the diencephalon, which is the caudal part of the forebrain. Again, we have the mesencephalon, and this is the area that doesn't divide. And then we have the metaencephalon, which is the rostral portion of the hindbrain, and the myelencephalon, which is the caudal part of the hindbrain. And here we see the adult brain structures that they develop into. From the teleencephalon, we get the cerebral cortex, the basal nuclei. From the diencephalon, we get the thalamus, the hypothalamus, and the epithalamus, which is also known as the pineal gland. From the mesencephalon, we get the midbrain of the brainstem, and from the metaencephalon, we get the pons and cerebellum, and the myelencephalon contributes to the medulla oblongata. And so again, this would be the area of the cervical flexure, and so above that we have brain, and below that spinal cord. Now, throughout all the brain sections, there are some cavities or canals, and in the teleencephalon we have 2 lateral ventricles and part of a 3rd ventricle. So that develops from the teleencephalon, and this 3rd ventricle also develops from the diencephalon. That ventricle forms a bridge between the 2, or is located between the 2. And the mesencephalon has what will become the cerebral aqueduct, and from the rhombencephalon, both its portions, we get the 4th ventricle. And these canals continue as the central canal through the spinal cord. So list the secondary embryonic brain vesicles and their cavities. And I just discussed those. So this would be an appropriate matching question, or perhaps a which one is the wrong answer kind of question. So for that, for example, we had myelencephalon contributes to the third ventricle, then you would know that was the incorrect answer. So make sure you know which of these become which, and be prepared for any variation of a question on those. So to dive deeper into the development of the brain vesicles, we'll look at each of the sections individually. And so we'll start with the rhombencephalon, and in particular the myelencephalon, which, as I said, is the caudalmost part of the hindbrain. This will give rise to the medulla oblongata, and because it's the caudal portion, it does continue directly into the spinal cord, and its central canal, that cavity running through it, will become part of the fourth ventricle. And the alar and basal plates remain to divide the medulla into dorsal sensory region and the ventral motor region. And as far as alar and basal, I don't believe we've discussed this in class. Let me draw you a quick diagram. That's a terrible drawing. Just ignore that one. You have these alar and basal plates, which are separated by this little groove called the sulcus limitans, and the top plate is the alar plate and the bottom one is the basal plate. And there's actually a really good diagram of this in figure 18.6, and you'll want to review alar and basal since we haven't discussed that in class yet, but I believe we're going to. So you'll want to review that on your own. And so continued growth of the alar plate causes it to migrate and become lateral and basal. So here we see a figure similar to the one we already saw, so I'm not going to discuss it too much. The only thing that's important here is the level of section B, which will be the next figure. And so the next figure will be a cross-section taken right here at the cervical flexure. So here we have the central canal right in the middle, running right through the brain. And the neural canal of the neural tube forms the central canal. Down at the bottom we see the pyramids, which are composed of corticospinal fibers descending from the developing cerebral cortex. And these two nuclei, the gracile nuclei and the connate nuclei, and nuclei plural because there's one on each side, these are both formed from the alar plate. And here we have a diagram of the alar plate and the basal plate, and you see them separated by this groove, the sulcus limitans. And some of the neuroblasts from the alar plate will migrate ventrally and form the neurons of the olivary nucleus, which we'll see in more detail in just a bit. And here we have the fourth ventricle again because it's at the level of the rhombencephalon. Okay, and here we have a further in time development of that same section. And we see that the alar plate, which is a light blue, has already begun to differentiate into some of the different somatic and visceral components. And the dark blue, which was the basal plate, is differentiating into the efferents. Here we see the olivary nucleus that we just discussed. and the choroid plexus up at the top. We'll see more of the choroid plexus in a while. Some of the functions you probably remember these from physiology, of the special somatics and general somatics. For example, this special visceral afferent, which would be this one right here, that forms the neurons that carry information about taste from the tongue to our brain. The general visceral afferent, that carries information on the subconscious sensation from the organs. So that would be stuff like blood pressure, the level of carbon dioxide in your blood, for example, maybe the stretching of the stomach, and so just different subconscious, in the sense that we're not really aware, normally at least, of, for example, what our blood pressure is, but your body has to, at some level, be aware of these things in order to provide homeostatic control, and that would be handled by the general visceral afferent nerves. Another example, the special visceral efferent, this one's right here, those carry the innervation of the muscles produced by the pharyngeal arches, and so, for example, the muscles of facial expression. And so all of that took place at the level of the myelencephalon, which becomes the medulla oblongata. Okay, the metaencephalon is the rostral part of the hindbrain, and that becomes the pons and the cerebellum, and that also contains within it the fourth ventricle, and the cerebellum is derived from the lateral expansions of the alar plates at that level, which end up fusing in the medial plane. So again, we have this familiar diagram, this bump right here, looks like a dorsal fin or a hunchback. That's a developing cerebellum, and we're going to take a look right through here, developing pons at the bottom. So this level of section B, so going back, we have the developing cerebellum not only forming this bump upward, it looks like it's right in the middle, but it's not. If you look at the section, there's one on each side, and they're both growing upward, so dorsally, so it's kind of forms this V shape. I guess it's more like a U shape. And so the lateral expansion of that forms a developing cerebellum. Here we have again some more of the afferents and efferents. Here's the fourth ventricle. Again, that's a continuation of the fourth ventricle that we saw in the myelencephalon, because as we saw in the figure that started off with the neural tube, the fourth ventricle extends from the metaencephalon all the way to the myelencephalon. Okay, here we've got, let's see. Okay, this is at 6 weeks of development. And so, there's a pointer. Here again is that developing cerebellum. We have the choroid plexus being formed here as the capillaries infiltrate this area. And the choroid plexus secretes ventricular fluid, which, combined with other components from other areas, forms the cerebrospinal fluid. And the particular component produced here is the ventricular fluid. So, as we said, the cerebellum is derived from the lateral expansions of the alar plates, and that mostly coordinates or is involved in highly coordinated automatic activities. So things that we can do, especially with practice, but don't necessarily do at a conscious level, like athletics, playing basketball, swinging a bat and hitting a baseball, riding, balance, stuff like that that you kind of have to learn, well, except for balance, but then are able to do automatically, and involves a lot of coordination. And we also see the telochoroidea, and in the adult that will form the medullary bellum. So this is at 17 weeks, and we see that, going back just a level, the fourth ventricle has tightened up and closed into this cerebral aqueduct. It's really small now, and so it's more of a canal than like a large ventricle. And there's various, I keep losing the pointer, there's a dentate nucleus and various other sections that you're welcome to learn on your own from the lecture. Okay, name the parts developing from the rhombencephalon. Again, we have the pons, the cerebellum, the medulla, and the fourth ventricle. So now we move on to the mesencephalon, which is the midbrain, and that does remain as a single structure. Both the forebrain and hindbrain split into two. This one doesn't. And decontinuation of the fourth ventricle from the hindbrain becomes a cerebral aqueduct in this section, and the alar plates become the tectum, and the basal plates become the tegmentum. So again, our familiar figure, we'll be looking at a section right through the midbrain flexure. So here we have the cerebral peduncles. These are heavily myelinated descending pathways. We've got the primordia and the substantia nigra, which is actually really important. It's all really important, of course. But this is called substantia nigra because, although it's shown in a light color, it produces a lot of melanin in these cells, which stain it black. And so it's known as substantia nigra, nigra meaning black. And these neurons, we certainly see the effect of problems in. The neurons in the substantia nigra inhibit the activity of cerebral nuclei by releasing the neurotransmitter dopamine. And if these cells are damaged or the neurons secrete less dopamine, the cerebral nuclei become more active. And since these are inhibitory and you're inhibiting an action, if these stop acting, that action becomes more pronounced. And that occurs in Parkinson's disease, which is exactly, this is the anatomical and molecular problem with Parkinson's disease. People with Parkinson's have difficulty starting voluntary movements because the opposing muscle groups won't relax, and they have to overpower those muscle groups. However, once a movement is underway, every aspect must be voluntarily controlled through intense effort and concentration. And here we have another cross-section this is at 11 weeks. So we see the cerebral hemisphere starting to form, and we're going to take a look here at the brainstem level. The first one will be section D at the level of the inferior colliculus, and then we'll look at section E at the level of the superior colliculus. So again, we've got the substantia nigra there, our pyramids. The substantia nigra has changed and migrated from that little circle that we had before to this level at 11 weeks, sitting right above the pyramids and just kind of stretched out along that length. Here's the inferior colliculi, one on each side. Those connect to the medial geniculate nucleus of the thalamus and are responsible for auditory reflexes and hearing. We've got the cerebral aqueduct running right through the middle there. The superior colliculus is much the same as the inferior colliculus, but this connects to the lateral geniculate body of the thalamus and is responsible for visual reflexes and information. Here we've got the red nucleus, which, like the substantia nigra, is more a descriptive term than anything else. The red nucleus is rich in blood vessels and so appears very red. Biologists aren't always creative. So now we move on to the forebrain, the prosencephalon, and its two sections, and we'll start with the diencephalon. The diencephalon gives rise to the thalamus, the hypothalamus, and the epithalamus, which is the pineal gland. And at this level, we don't have differentiation between alar and basal plates. The cerebral aqueduct continues up into this level as the third ventricle, and part of the diencephalon contributes to the formation of the posterior lobe of the pituitary gland, which is the anterior lobe formed by Rathke's pouch, and we'll see the development of the pituitary in a little while. So this particular portion that it's referring to would be called the neurohypophysis. So here we have our normal fifth week diagram, and then we see the same section at 7 weeks. The cerebral hemisphere has gotten much larger, and the brain is starting to bend. We see the origination of two of the special senses, the olfactory bulb and the optic nerves. And so the telencephalon begins to move out, and the diencephalon will be left behind. So right here, this is okay, I guess this is at 7 weeks also. So this is just a cutaway from panel B. And we see the three sections right here, the epithalamus at the top, the thalamus in the middle, and the hypothalamus down at the bottom. And these all begin to form in their dorsal ventral orientation. And so just as a reminder, because we're in the brain, once the cervical flexure forms, you get a different orientation of where ventral and dorsal are. So you're sort of working with two coordinate systems. We'll draw a happy guy. And so in the body, you have dorsal being towards the back and ventral being towards the front, towards the chest. In the head,